biology, but don't be afraid. As last time, it concerns pretty much that you get a feeling of the role of membrane proteins in the functioning of a cell, and I hope that this knowledge will help you also in, for instance, studying biological membranes or in designing membranes for particular applications, for particular purposes. And the topic of today, the first session, would be the origin of membrane bioenergetics. So what we want to understand is, how do membranes help that a cell is supplied with energy, and that's typically in form of ATP. How is ATP generated in a cell, and what is the role of membranes in this process? So let's start maybe to go back in time. We go now very, very long back. Not only 100 years, 1,000 years, million, milliards of years we go back. At that time, the Earth was basically formed, cold enough that water would condensate, that we would have liquid water. We would still have some volcano activity, and maybe it would look like that. And you see here, for instance, a place a little bit on volcano stone, a little bit with sulfur, and the atmosphere would contain carbon dioxide, water, carbon monoxide, nitrogen, ammoniac. There would be no oxygen. But that was the time when life started. So all life started actually on Earth without the presence of oxygen. And you can maybe, imagine that these type of rocks, when they are from the volcano, they are very porous, like a sponge. Well, if you have at home maybe a sponge, there's a little channels in. So this material has a lot of cavities, small cavities. And maybe in such type of cavities, the organic material that was there in the ocean started to undergo simple reactions, started maybe to form a layer of lipid, starting to make the first barriers, so that in a cavity you would have a closed space which would almost mimic like now a cell. And that's the basically idea how life might have started in terms of getting separated from the environment and basically to start to use organic material that at that time was very rich. You see also the sky was not really blue, oxygen was not there, so no ozone layer. So it was actually more a type of a sky that you would find maybe on the Venus-like, so very cloudy and not so beautiful. When we look now, this morning was beautiful weather, you see a beautiful sky. But life started, and maybe the very first cells that you can think about would have now this type membrane that we discussed, not necessarily a bilayer that would have basically two independent lipid layers, but maybe you would use these type of lipids even that are connected, where the tails are connected, these very strange lipids that we are calling border lipids. And what would need this organism to live? It would need to take up substances and would use these substances to actually generate ATP. And to do so, it probably would also very early need a type of facilitator and uptake. And we talked about that this could be secondary uptake systems that are driven by a gradient. So most likely, these first organisms would have a mechanisms to extrude protons and use this gradient of protons to move organic substance in, to accumulate organic substance. So that could be the first organism that we think. Maybe they even use this proton-driven transport not only to uptake material, but also to actually secrete material like toxins to protect themselves in this ocean around with all these organic substances. Now, when an organism would do this, it would be, of course, very costly. So it would basically take up organic material, would do glycolysis, would use the ATP to create this proton gradient. So it would actually use quite a lot of energy just to take up. So organisms that would basically came up with a system that would not use ATP to create a proton gradient, but would use energy-rich electrons that are also produced during the process of glycolysis, would use these electrons instead, they would have an advantage because they could use the ATP for other processes. So probably another thousands or millions of years later, some organisms came up, or at least one organism came up, with a proton pump that would be not driven by ATP, but would be driven by electrons that are derived from the organic material that they would have taken up. So these organisms would have a huge advantage now, because they would still make a proton gradient that can drive uptake, but would not waste ATP, but rather would use the energy-rich electrons. Okay? And if such a system would be very efficient, so if basically these electron-driven proton pumps would very efficiently use the energy that is delivered from electrons from the organic material that is degraded, you would build up like a cascade of proton-driven pumps, so you would basically be able to make a very strong proton gradient that then could be used not only for the uptake of material, but allow basically the ATPase to work reverse. Remember we talked about yesterday about ATP-driven pumps. There are 3 types, and there was basically one example, the F-type and the V-type ATPase, where the F-type works reverse. So that would be a reverse-working ATPase that now makes ATP. What a wonderful organism. It can fully use all the energy that are in the nutrients that are taken up to generate a strong proton gradient that can be used to make ATP and on top can be used to take up organic material. So these bacteria, they would best compete with all the other bacteria around. They would basically, that would be a big bang in evolution. Those organisms would now start to occupy the whole ocean. Yeah? And you see, so the principle here is without a membrane, such a system would never have been able to build up in terms of separating the cell from the environment and in terms of basically allowing the cell to drive the metabolism, to generate ATP, to take up material for degradation and for synthesis. Okay? So probably another million years, the world was very happy with these organisms, and what they do? They would not eat up all the organic material. The ocean would be full. But of course, if we consider this, at a certain moment, the organic material would be then limited. No? So these organisms would start to compete for the organic material, and that gives a lot of pressure for organisms that maybe find now a new solution to actually generate ATP. And what happened was that basically with the purple bacteria, organisms appeared that can use the energy of light. That was, again, a major development. But the prerequisite was that they have membranes to be separated from the environment and to use basically these membranes to generate gradients of ions, for instance, protons, across the membrane. So these bacteria, they use the trick. So they have basically a system, a light-having protein system, that steals electrons from sulfur hydrogen. That's basically a substance that is, the electrons are not easily available, and then they are energized by the light, so they get energy-rich electrons, and these energy-rich electrons, they can drive a proton pump. So a light would be used to energize electrons taken from anorganic material, not from organic material, from anorganic material now, to energize a proton pump, and this proton pump would be essential to make ATP via these F-type ATPase and to take up material. And that means these organisms would become now dependent on light, but not anymore depending on these organic substances. They can basically use light to drive their energy metabolism. And you see basically now when you have these components, that is a proton-driven pump that already pre-existed in the other bacteria. It was now only newly combined with a system that can harvest light. So nature always used the components that are already there to reassemble. It's like on the level of the lipids we talked about it, on the level of proteins we talked about it. Now we talk even on the level of protein complexes with the same principle again. Then, probably another million years further, cyanobacteria came up. And they basically found another solution to steal electrons. They were able to steal electrons from water. They could split water. That's something we would like to do now. No? You know, this energy problem on the Earth, so you would like to basically make endless fill. So if you would be able to split water, you get oxygen and hydrogen. And hydrogen and oxygen you can burn again and you would have a lot of energy. So these bacteria found a way to split water, which requires quite a lot of energy. So they developed a light harvesting system that is able to use the energy of photons to steal the electrons from water, to split water. As a byproduct, you get oxygen. It's a waste product. That was a disaster, no? For all the other organisms, because oxygen is very aggressive. You notice, for instance, if you have metal, iron, if you wait long enough, it will be oxidized. And you can imagine that organisms that are living in an unorganic atmosphere are now get exposed to oxygen. They don't have any protective mechanism. This oxygen might attack lipids, the double bonds in the lipids. The oxygen might attack the proteins, the amino acids. So for several organisms, at that time, these organisms were not dangerous in a way that they produced the toxic byproduct, oxygen. All this oxygen first accumulated in the water, but at a certain moment the water was saturated and our atmosphere started also to get oxygen. Okay, so these particular organisms were able to actually take electrons from water, to split water, to energize it, to use basically in these electrons to drive again a proton pump, to make a proton gradient. And then they even had a second system that could re-energize the electrons once more to be transferred to the reducing substance. And that is important for metabolic processes. We don't go in detail here. That's more for the biochemist. But they could basically energize the electrons to drive proton transport across the membrane and to re-energize the electrons to give basically new chemicals that are important into metabolic pathways of the cells. So these cells could completely live now on light. Phototrophic organisms. Okay. So now we have basically here these evolutionary steps, and I talked about that now with the appearance of basically cyanobacteria, our atmosphere got oxygen. And that allowed to develop basically another type of cell that could use oxygen. And those cells would have an advantage because those cells would oxidize now organic material very efficiently down to carbon dioxide. Normally, when you do for glycolysis, for instance, you still end up with an energy-rich substance. It's not completely broken down. You have, for instance, glucose, and you end up with pyruvate or lactose or ethanol, depending on the organism. It's still energy-rich. No? I mean, ethanol you can still burn, pyruvate you can still use. But if you can further oxidize it now with the help of oxygen, you get the maximum of energy. Actually, you almost burn it. You burn it in a cold way in the cells of our body, and you can get a lot of energy. So these basically cells here, they would be able to use the oxygen and take electrons from the organic material. They would live from organic material and then transfer these electrons down basically to the oxygen to form water. And while they do this, they would again drive proton pumps to generate a proton gradient that then can be used to make ATP. So in all cases, you have basically always a membrane involved that is required for the basically transmission of the energy of electrons into the later formation of ATP. Okay, So if we now consider this in terms of evolution, so you would basically have here an organism starting as a single cell, and maybe this cell, this organism at a certain moment was able to generate quite a lot of membrane. The more membrane you have, the more processes you can basically let go on, the more proteins you can embed. No? So this organism here already would be a type of cell that would still live from organic material, no photosynthesis, nothing like that, but would have a high capacity of producing material and to break down material due to the large amount of membranes already in. And then an important event happened. So this cell, the so-called pro-eukaryote, took up another bacterium. But instead of eating it away, it made a friendship. Instead of basically using this bacterium and to digest it, it started to make a friendship. And that basically ended up with the first type of heterotrophic cell, a type of precursor of the animal cell that then went to animal cells. So those cells, part of these organisms that were able to basically now harvest the energy or to share the energy that is produced by this bacterium inside, they basically then took up another bacterium, cyanobacterium, that became a plastid. So now they had a chloroplasts. So you have basically now from the original cell that was an organism using only organic substances, you had basically now an heterotrophic eukaryote that was able to produce ATP from organic substance by the use of oxygen, and you would here have a photosynthetic organism that would be able to produce basically ATP by the use of light. So that's the way how basically in the end our cells, the precursors of our cells were formed. And then this cell gave later evolution to all the plants that we know now. This cell gave then rise to all the type of heterotrophic organisms, animals that we know. Okay, so what is common in all these? Common is, it's the membrane. The bacteria, the plants, the animals, they all use the membrane, a bilayer, in which basically a proton pump, a protein is embedded, or a protein complex is embedded, that can use electrons to pump protons across the membrane. That allows to generate a proton gradient, and this proton gradient is then used by a second transporter complex, by a second membrane protein transport to generate ATP. That's common for the mitochondria in our cells, and those two protein transport complexes would be localized on the inner membrane of the mitochondria. And in the chloroplast, it would be localized here in these thylakoid membranes. So just to make sure that we are all on the same, so this mitochondria, why would you have the mitochondria two membranes? What is the reason that in ours, basically the mitochondria have two membranes and not only one membrane? Because we would have started with a bacterium that would be have taken up but not digested, but stays in the cell. So where does basically this membrane comes from? The outer membrane. Where does it come from? From? It's from the host cell. It's almost from the plasma membrane. This membrane is a plasma membrane-like. And this is the membrane of the original organism taken up. What would this tell you without much knowledge about the lipid composition? What would you expect for these two membranes in terms of lipid composition? Very different. Very different, because one is a foreign organism that you take up, but you don't digest, but you encapsulate it with your own membrane, which comes from you. So this membrane would be most similar to the host cell. This membrane would be very different. Okay, now this is not maybe well drawn here. You have here the basically chloroplast with the thylakoid membranes. You have an inner membrane and an outer membrane. Why do we have there 3 membranes? Maybe we go back here. Oh, sorry. Now we have an organism with 3 membranes, quite complex. This is because the starting organism already had 2 membranes, was taken up, engulfed but not eaten up, which means if you think about the membrane, the outer membrane, what would be the lipid composition? What can we expect from the lipid composition of the outer membrane? Would be most of the host cell. What can we expect for the lipid composition of the thylakoid and the inner membrane? They would be more similar to the cell that have been taken up. Yeah, you see? So and indeed it is like that. So if you would basically, and that is actually also support for this endosymbionten theory where people came up with, you can actually do this also on the biochemical level. The membranes are different in terms of outer membrane, inner membrane, for the chloroplasts and for the mitochondria. Okay, good. So now let's have a look a little bit more in details to the membranes. So this would be now the mitochondria. We have here the outer membrane and we have the inner membrane, and you see that this inner membrane is so typically invaginated. This is also what you knew now already from the school when you would draw. The mitochondrium in the drawing is most easily to detect because the inner membrane is quiet. No? And what is the reason for that? You would typically what say, why would this membrane maybe, you know, in evolution be so invaginated? What could be reasons for that? You want to give me some reasons? Surface, that would be the typical answer. Thomas, this is, we need a lot of surface. When we have a lot of surface, we can put a lot of these transporters in the membrane. So we have a very efficient system, right? Let's see whether this is the whole truth. There is some truth in that, but not necessarily all. And we have basically here the chloroplast, and sorry for the drawing, so we have basically the outer membrane, inner membrane, and the thylakoid membrane. Now the machinery that makes basically this gradient and produces ATP is sitting here on these thylakoid membranes. Why would Thomas have drawn this green? Is there any reason to give this a green color? Chlorophyll. So, chlorophyll. Very interesting molecule. And this chlorophyll is really interesting because it's bound to a membrane protein, right? And this membrane protein is then basically pumping protons, right? So it's the light harvesting part, which basically takes the energy of the photons. And this energy of the photons, it's transmitted into the movement of the protein that starts to pump protons. Okay, so in a way, it's not absolutely true. The inside is not green. It's actually the protein that is in the membranes that is green, right? But since these structures are very small, you can just see green, no? And also from the leaves, you just see them green. Okay, so now there's one important aspect. Okay, here we produce basically a proton gradient on one membrane, and we have a membrane that is basically around it. Here we produce the proton gradient in the lumen. So obviously in the evolution, something went differently in the terms of mitochondria and in terms of the chloroplasts. So let's have here a look. I need to tell you that this membrane here, the outer membrane, it's quite permeable. It contains pore proteins, proteins that make really large pores. The consequence would be if you pump protons here into this mitochondrial space, the inner membrane space, the protons have a good chance to leave the mitochondrium into the cytosol. That would be a disaster, right? Because we put so much energy using the energy of electrons to put now all the electrons on that side, and then they disappear. Actually, in fact, this is wonderful here. You can see this is an outer membrane mitochondria. Yeah? A lot of pores. So now we have here basically the inner membrane. Now we pump protons here over with energy-rich electrons. In the case of mitochondria, it comes from organic material. So we pump them all here, and they would start to diffuse. And they would go away. But in fact, we want to have the protons here to be used by this F-type ATPase to leak back and to drive ATP synthesis. So from the construction, this system is not nice. So actually, somehow, as a type of engineer, there's a failure in this. Right? In case of the chloroplast, it's different, the situation. There you pump the protons into a closed lumen. They're all here. And they cannot escape unless they cross the membrane again via an F-type ATPase to make ATP. And due to this problem, this drawing here and this construction is also not absolutely correct because the invaginations are very tight, also very, very thin. Yes, you have a large surface, but what do you see now? You make almost a separation here from the space, the inner space. So all the protons that you pump, they accumulate. Yes, they might have a chance to diffuse, but diffusion is limited. So they stay quite here and can be immediately used by the ATP synthase to generate ATP when they flow back to these proteins. So these invaginations, as one hand is to make a large surface, but the second thing is also to limit the diffusion of the protons that have just been pumped by the electron-driven proton pumps to the basically mitochondrial space. Okay, so these constructions, they have a biological, actually, also physical type of reason to limit the diffusion, to maximize the use of the protons. Okay, so now you have basically seen the basic mechanisms, and we go just a little bit more into the idea of how to use a membrane to make a gradient, because I told you that in evolution, not only one electron-driven proton pump developed, but actually several, and they teamed up. So what you see here is now one proton pump, a second proton pump, and a third proton pump. And they are all in one membrane. And they all use the energy from one electron. So the electron, in this case 2 electrons, are delivered to this proton pump, which, when you have an energy-rich electron, that starts now here to go into this protein complex. And while it is walking to this protein complex, you will change your conformation. And this change of conformation will allow a proton to pump. Now, this electron is still not completely energyless. It can go to the next proton pump, which also triggers now a movement and pumps protons. It's still not lost all of the energy. It still has enough energy to even drive another pump. So you use the energy of 2 electrons very, very efficient by allowing these electrons to pass 3 complexes that all pump protons. So you generate a strong proton gradient by just taking 2 electrons from an organic substance that you break down. Okay? So for historical reasons, they have now not numbered exactly like you would expect 1, 2, 3, but 1, 3, 4. But this is something for those that really want to dive into the details. The principle is 3 proton pumps. And in between, you see you have also helper proteins or helper molecules that help in the transfer of the electrons. Okay, just since we are talking about membranes and we're talking about membrane proteins, if you want to design this in a real way, not like it's basically there drawn, I think I would like to have your help again. Huh? Yes, both, because we need 3, no? We need 3 persons here. We would be 3 proton pumps. No? We would be very happy, because whenever an electron comes,
you would basically pump a proton. No? And we would do this, pump a proton. The first electron, so the two electrons come to you. What is your movement? Yeah, we are in the membrane, and we basically pump in this direction, make the movement. Oh, like this, yes, you are right. Yes, so now we do it, so okay. Electron comes, you, action. Electron comes to me, action. Electron, no, you not. You gave, no. Electron comes to you, okay. You get your new electrons. Then I get it. You get your electron. You get your electron. Okay, but we need helpers, no? Tanmay, you want to be one helper? If you go a little bit more distance, so you would be, for instance, you could be a soluble helper. You are in the membrane and you have this fantastic ability to take electrons. So you can go on his side shortly, take the electron, bring it to me, and then I do this. And then we would have here another helper, and then you would do that. Okay, now we do it stepwise. And the drawing is like that. You are the helper. You can always like this. And you are always busy. Yeah, you are always busy. You walk, and whether you have electrons or not, you walk. Sorry for that, but you walk. You walk. Okay, now go ahead. You get electrons. Yeah. First this. Yes, then you give an electron. Zack. And then we have another helper here. Zack. You still walk. Do you still get electrons or today not? Where are your electrons? Okay, I have more electrons. Yeah, you get more electrons. Good. Okay, thanks, Tamar. Stop. Wait. Do you think it's like that? Is this a good way? Because that would be the drawing implying, right? Would this be? Have you watched Tanmay here? Can you pretend as if you would have electrons? Okay, thanks. Would it not cost time? And it's a membrane, right? So if we would have a two-dimensional, what else could you do on movement? So now, walk, pump, get electrons. Yes? How did you know that he has no electron? Yeah, you would always walk, and this would be not very efficient, right? There would be a delay. Do we want this? We want to be good. We want to be fast, right? What would be a solution? What are solutions you could come up? What do you think? To make this more efficient, this process. Do you have some fantasy? We always need students with fantasy because sometimes, you know. Okay, yeah, that could be an option. But what if, I hope you are not, it's okay with you, Tanmay, and your colleague comes also. Would this be not cleverer if we come closer together? If we come very close together? And would Tanmay need to move no much? What would your movement be for the electrons? Ah, more, faster. Yes, good. What is about you? Do you want to have this? Yeah. So how do we call this now? A protein complex. That would be, so actually, in fact, you know, what this drawing is just to illustrate a little bit that you see there are proteins. But in reality, they are very close to each other. It's a super complex that we are talking about. We are actually sitting so close together that Tanmay as a partner would not need to diffuse your, you know, endless around. You would be in this complex very fast. And you would be also less stressful for you, you know, to walk around. So we would be super efficient in this. And that's what's in the reality is. So these proteins are super, super packed. No? Okay, thanks for that. That was a little illustration that this drawing is actually misleading. It serves a good purpose to explain each step, but it doesn't reflect when you would basically put these proteins in the membrane, they would form a complex.
very tight complex. Okay, good, that we have understood. So now, basically, okay, we could put this in a membrane, we could basically give a substance that delivers the electrons, and we would get proton pumping. So we could build it in a vesicle. And then we could start pumping basically protons. So we would then get in the system either an accumulation of protons in the vesicle, or you would move out depending on the orientation, how you have inserted this into the vesicle, right? Yes? Much. There's basically maybe one lipid in between or so. There is hardly any membrane between them, but they sit in a membrane. But the proteins by themselves, they form a super complex. So this drawing, it's more like to understand the steps, but it's not the situation in terms of topology, how the system would really look in the membrane. Okay, so now we have a proton gradient, and what else would we now need to get ATP? Which type of protein would we need to include in this complex? Exactly. So we would need now this type of protein complex that would use the proton gradient to make the ATP. Okay, so now you see how a membrane can be used basically to generate ATP by protein complexes that are on one hand generate a proton gradient, and by a protein complex that uses this proton gradient to make it. Okay, so like very briefly, remember the idea of this transport complex was it works like a turbine. So the protons, basically, that the proton gradient that is generated, the protons would go back through this protein complex, and then one part of the protein would get in rotation. And this rotation would basically affect the conformation of this F1 protein complex. It's in 3 subunits that can bind ADP, phosphate, make the bonding, and you get ATP. And it does it basically in a cycle. You have 3 subunits. Each of them can bind the ADP, the phosphate, and can link it. So in fact, when you have 3 and you have basically now the protons flow, that would give me a rotation. And if I have 3 subunits, we could still play this actually. You want to be a subunit, Tanmay, and you want to be the other subunit. You can turn to this side here. Sorry, you can turn your face to the audience. Yes, like that. Turn a little bit more. Yeah, like, okay. Yes, you can be a subunit like that. You also want to be a subunit? Yes. Then we all move a little bit more like that. So, and you are the other one. So you turn to this side shortly. So what they all can do is they can bind ADP, phosphate, combine it. But they would only do this type of literary movement. No, I don't. So no, I do this, but it's not true. I do this. And then Tanmay can change basically the conformation. No? You do your own job. You make ATP, you make ATP, you make ATP. No? So suck, and I get dizzy, but the transporter doesn't dizzy. And this turning is because of the proton flow here on my legs. The proton flow drives me crazy, while I basically always make these subunits here busy. Okay, thanks a lot. And then we have our ATP. That's what's going on. Good. Now, wonderful, all fine. But what happens? Where do we get the ATP? We get it produced inside. It's all inside of the mitochondria, in the matrix. What a disaster. Because where do we need the ATP? We need it outside. We need it in the cytosol. And ATP is not permeable. Why is it not permeable? Why can ATP not cross a membrane? What were the three things you should then think about? The 3 criteria. When is a substance permeable or not permeable? Size, polarity, charge.
What is it about ATP? Large. Of course, there are larger molecules. Proteins would be larger, but it's larger than an ion. What else? Charged. 4 times negatively charged. Super negative molecule. A super negative molecule, ATP. 4 negative charges. One of the most negative molecules, no, if you think. In biology, at least. In biology. So this would never leave a membrane. It will never cross a membrane. So you need here again a transporter, an exchanger. You see? And this would basically, this exchanger is an antiporter. It allows ATP to go out and ADP to go in, because the ADP is needed to form ATP. No? But what do you see here? What is the charge of ADP? It's -3. That's the charge of ATP -4. What would this mean? If you exchange a substance that is basically 3-fold negatively charged against a substance that is 4-fold negatively charged. There's a charge imbalance, right? It's not equal. It's not minus against minus. It's 3 minus against 4 minus. But in this case, you see the proton gradient. It helps actually to get in the ADP and to get out the ATP. So it's a wonderful system, this antiporter. No? And of course, now you can see you need even other, basically, transporters for other material to go into the mitochondria that would then help to deliver the electrons in the end. No? You would need, for instance, in this case, an organic substance pyruvate that is further broken down, delivers the electrons that are then used by these electron-driven transporters. So this inner membrane, it's basically comes from a very busy organism in the evolutionary time. So it's an organism that was able to generate ATP from a proton gradient using organic material. And this we still carry in us in a way that we became, our cells became friend with this organism. Okay, so this is just a summary here that you see that basically in evolution, nature found similar solutions for a problem. How to make ATP. In both cases, nature uses membranes, a lipid membrane. In both cases, nature uses proton pumps that generates a proton gradient. And those basically gradients are then used to drive an ATP synthase to generate ATP. In the case of the mitochondria, the electrons come from our nutrients that are broken down. And in the case of chloroplasts, the electrons come from the water that is splitted by the help of light. So those organisms can basically produce ATP just from light. Those organisms carrying mitochondria can produce ATP only from organic substance. So how is it now, how would it be in a plant when there's no light? They will use the mitochondria because plants, remember, they have mitochondria and chloroplasts. If there's light, they use the chloroplast. If there's no, in the night, they still need to make ATP. But they can do this with the help of the mitochondria. So part of the substance that they have produced during the day need to be broken down again to sustain, of course, the life of the plant during the night or when there's very dark periods. Yeah, with this, I would like to end. Just to remind you, probably the first membranes were basically used to allow the generation of proton gradients. And these proton gradients were basically the basis to generate ATP and to allow the uptake of substance by so-called secondary different active transporters. And the formation of these membrane structures was basically the prerequisite that you could have an independent organism. And the evolution of the electron transport chain was basically important for efficiently use the energy of electrons to generate a proton gradient. Remember, we had not only one, the early organisms maybe only had one proton-driven, electron-driven proton pump, but then the later organisms, they actually were able to have
a chain of membrane proteins that use electrons to pump. And that we call a respiratory chain now, for instance, in the mitochondria of our cells. Yeah, that would be for today for bioenergetics. So we have a very basic introduction into bioenergetics. You have seen how important are membranes in this process. We will then later, after the break, come to a new topic, how are membranes used actually in signaling processes. Any questions for this part? Yes, please.